Hi, welcome to my talk. I'm Meike Hettwer, and I'm very honored to be here and present to you my work, Coordinated Cortical Thickness Alterations Across Mental Disorders, which is a transdiagnostic ENIGMA study. So one fundamental question in understanding the brain basis of psychiatric disorders is to what extent it represents disorder-specific or transdiagnostic features. In our project, we are taking the angle of cross-disorder similarities, which should be considered complementary to disorder-specific research. We know that there are overlapping risk factors among diagnoses, like shared genetic risk variants, but also non-specific environmental risk factors, such as stress and trauma. And these shared risk factors may be one reason for the phenotypic overlaps we observe at the level of brain neuroimaging. For example, as seen in transdiagnostic meta-analyses, but also when spatially correlating disease impact maps. But effectively, these alterations span multiple neurobiological levels. So it's a central question, what drives the cortical patterns that we observe to be in part shared between disorders? And among others, it seems to be the underlying network organization. So regional effects are not independent, but interrelated and thus potentially coordinated. But it's still unknown whether also transdiagnostic effects are coordinated within a shared network across the cortex. So the efforts of openly shared data from worldwide consortia such as the ENIGMA consortium, it becomes increasingly possible to investigate phenomena across clinical diagnoses. And that's also what we aim to do here. So for this project, we accessed Cohen's D-maps on cortical thickness from 12,000 patients with 6 mental disorders through the ENIGMA toolbox. And we included bipolar disorder, ADHD, autism spectrum disorder, major depression, obsessive compulsive disorder, and schizophrenia in our analyses. And we also accessed functional and structural connectivity data from the Human Connectome project for our contextualization analyses from the healthy young adult cohort. With the aim to find systematic patterns in cross-disorder illness effects, we generated a co-alteration matrix representing correlations of illness effects. So basically correlations of Cohen's D values between regions and across disorders. We then identified co-alteration hubs by taking the sum of strongest connections, showing us regions where illness effects are strongly coordinated with other brain regions. And these hubs were correlated with normative functional connectivity hubs, indicating that co-alterations are not randomly organized, but follow underlying connectome organization. We next aim to see whether there are factors contributing to this pattern and checked whether there are structures that are strongly connected to co-alteration hubs and may shape the cortex-wise pattern as potential disease epicenters. So we used seed-based connectivity profiles from the Human Connectome project data and systematically correlated that with our co-alteration hub map, correcting for spatial autocorrelation. So regions that are strongly connected to co-alteration hubs were identified as potential disease epicenters. We observed most prominent epicenters in ventrolateral prefrontal and mediotemporal regions. And there is an overlap between structural and functional epicenters, but functional epicenters emerge above and beyond, which is not necessarily surprising, given how resting state functional connectivity is computed and also includes spatially distributed and polysynaptic networks beyond hardwired tracts. So the first chapter of our analyses embeds regional alterations within cortex-wise transdiagnostic covariance networks. The fact that co-alteration hubs followed patterns of normative connectivity hubs indicated a role of nodal stress. So regions that are highly interconnected and thus highly engaged in many subnetworks are more vulnerable to pathology. So here, regions with strong co-alterations also seem to be regions being highly interconnected functionally. So nodal stress is the likely explanation for the co-alteration pattern we observed. And epicenters preferentially emerged in regions known to extend long-range connections, mainly heteromodal regions, facilitating their contribution to cortex-wise patterns. And there are also regions like the prefrontal cortex with protracted plasticity, which is needed for cognitive refinement, but also increases vulnerability during development. In a second chapter of our analyses, we aim to identify cortical axes along which co-alteration patterns were organized using a gradient approach. So gradients reflect the similarity of network or covariance embedding in a unidimensional space. they could be seen as a component of a dimensionality reduction. And similar colors indicate similar network embedding. So the principal gradient shows that frontal and temporal regions have maximally different co-alteration profiles, explaining 36% of variance. Whereas the second one segregates regions holding primary sensory and limbic regions from multimodal occipitoparietal cortex. We then did a cytoarchitectonic and functional decoding to understand what these gradients reflect. So we stratified the axes according to 5 cortical classes as defined by von Economou and Koskinas, which reflect differences in microstructure and cortical complexity. So our principal gradient segregated agranular from granular cortices. So basically thick cortex with large cells from thin cortex with many but small cells. So the cytoarchitectonic classes with most different microstructural complexity are also the ones with most distinct illness effect covariance, suggesting a certain structural guidance. The second gradient segregated granular from parietal regions, and together this tells us that areas with shared cytoarchitecture are more likely embedded similarly within pathological networks. We then performed a task-based functional meta-analytic decoding using the NeuroSense database to see which functional networks are segregated by the 2 gradients. For that, we merged the 2 gradients. So that's what we see here depicted on the brain. And also that reflects the color coding of functional terms. And we observed differential vulnerability in regions involved in basic sensory versus attention or perception related versus higher order cognitive tasks. So even though we know that these processes are interrelated functionally in many mental disorders, they're embedded in different pathological structural covariance profiles. Together, cytoarchitectonic and functional contextualization implies that the embedding of regional vulnerability is coordinated across the cortex, primarily between regions with similar microstructure and functional task engagement. And this may be due to similar local computational strategies supported by different wiring strategies, or also shared maturational sequences and the degree of plasticity. We also know that regions with similar microstructural layering profiles are more likely connected. So there may be a structural scaffold for co-alteration networks we observe. In line with the findings from cytoarchitectonic contextualization, the levels of functional engagement of cortices involved in similar tasks also appears to leave brain regions processing similar types of information with shared susceptibility. Together, our findings indicate that shared illness effects occur in a synchronized fashion and along multiple levels of hierarchical cortical organization. There are some limitations that I would like to address. For example, we match the ENIGMA groups by including adults, and maps are additionally age corrected, but the included disorders have a different age of onset, like autism spectrum disorder compared to schizophrenia, for example. So there is variability in disease and medication history, which was a bit of a trade-off with actual age or developmental differences. There is also certainly some within disorder heterogeneity, which we cannot account for here. And ENIGMA data is traditionally shared in the Zekin-Kiliani space. So we are left with rather sparse data points and also do not have other parcellations to replicate our data with. Overall, by investigating the embedding of regional vulnerability in a transdiagnostic context, revealed coordinated cortical alterations that are shaped by connectomic, cytoarchitectonic, and functional characteristics. I would like to thank the Cognitive Neurogenetics Group at the Research Center Jülich and Max Planck Institute for Human Cognitive and Brain Sciences, and also all ENIGMA working groups contributing to this work. Thanks a lot for your attention. I'm happy to take questions.